Good evening, everyone. So, how is your preparation going? Am I audible? Yeah, okay. So, today I will discuss Unit 4 Immunology with you all, right? So, immunology, as you have seen in your syllabus, it is quite big. It's a big unit over there. Like, it's a part of the unit, but itself is a size of an unit. And immunology, the best book to study for immunology is QB. Okay, so if you understand the things step by step, the immunology chapter or immunology section is very interesting. All right, so what I'll do today is I have selected just 5 questions today, not like how I always do 10 questions. Today I've selected 5 questions, 5 part C questions. I will discuss the questions and at the same time I'll discuss the concepts. I'll explain the concepts behind it. Okay, with pictures and all. So if other days I only give you the explanation of the question. Today I'll give you the explanation of the concepts also, so that it becomes a revision series also for you, right? But the rule is same. You have to try a question or try the question. So I'll show you the question, I'll discuss the concept, and then you have to try the question. Right? Are we all ready? Shall we start? Give me some responses. Come on. Yes, good evening, Rinku. I hope I'm audible. Like, if you don't give me responses, I won't even know that whether I'm audible or not. Okay. So, the first question. The following steps or events represent the pathway of presenting extracellular pathogen to cytotoxic T cells. Number 1, fusion of endosome membrane with the virus and escape of RNA and protein in the cytosol. Okay. Next, assembly of class 1 MHC protein with bound viral peptide in Golgi apparatus. Statement C, proteolysis of viral proteins by the proteasome. Statement D, binding of the peptide to alpha chain and stabilization of the assembly of alpha chain and B2 microglobulin. And statement E, recognition of viral peptide by cytotoxic T cell. Which one of the following option is the correct sequence of events? Okay, so you have to say which is the correct sequence. Now, this is a part which is there in your MHC. Okay, the chapter MHC, major histocompatible proteins. Okay, in that chapter, you will get the details of this question. So there you will have to study about the class 1 MHC, class 2 MHC, and how they process the antigens, which one is for the exogenous antigen, which one is for the endogenous antigen, how they process it, how they present it, who recognizes it. That entire details you will study in that particular chapter. So I have just taken a picture from that chapter, which somehow summarizes the processing and presentation of the antigens by the two classes of your MHC molecule, class 1 and class 2. Now, first one is talking about the endogenous pathway, class 1 MHC. Okay, so any endogenous pathogens or endogenous substance, it can be some proteins, okay, which is endogenous, like it is present within the cell. If that needs to be projected outward, all right, then you are going to project that through class 1 MHC molecule. Now here let me tell you, you have to study the structure of the class 1 and the class 2 molecules as well. Okay, you will get that in the chapter as well. Okay, Saritha has already given me an answer. Fine. Oh wow, Rinku has also given me an answer. That's super fast. Very good. But however, I'll still explain this, right, for the others who are new to this part, and then I will come up with the correct answer. Okay? All right. So class 1 MHC, if you see, you have this endogenous antigen. 1 minute. Let me see if I can zoom this for your convenience. Yeah. So endogenous antigen, okay, that means present within the cell. Now, first thing what happens is this antigen gets degraded. Okay. So the way of degradation is through proteasome. So this particular antigen, which is actually protein, right? So this will get ubiquitinated and it will be sent to the proteasome, where in the proteasome it will get degraded. Now these small peptides, they will be recognized by the TAP receptor, okay, which is present in your rough endoplasmic reticulum. All right, transported to the rough endoplasmic reticulum via the TAP receptor. So it gets in, all right. Now, when it gets in, then there are these on the other, like parallelly. So one side the peptides are getting in. On the other side, the MHC class 1 molecule is getting processed. All right. So we see this class 1 MHC molecule associated with calnexin. So calnexin, tapacin, these are all the chaperones which helps in the proper folding of this molecule. Okay. So this first happens with your calnexin, and then it is sent to your tapacin, okay, which is present close to your tap receptor here. The not okay, the transporter, not the receptor, sorry, the transporter. Okay, and it gets further folded. Okay, the final folding happens here with the help of your cal reticulum also. All right, and so now you have the beta 2 microglobulin chain also associated with the alpha chain. And when this processing is complete, then this molecule is loaded with the peptide. Okay, so now you can see the molecule loaded with the peptide, and it is sent out. Okay, it is excreted out of your endoplasmic reticulum. So class 1 MHC alpha chain binds calnexin, then beta 2 microglobulin. Calnexin dissociates, reticulin and tapacin binds. MHC captures peptide, chaperone dissociate. Okay. Now what happens? It enters the Golgi. Now, when in the Golgi, from the Golgi complex, it will be sent out through the vesicle. Okay. So, class 1 MSC peptide is transported from endoplasmic reticulum to Golgi complex to plasma membrane, where it projects the peptide outside. Fine. Yes, exactly. Alpha 1, 2, 3, beta 2, MSC class 1. Absolutely. Okay. Now that goes with the endogenous. Now coming to the exogenous. Exogenous means here the antigen is present outside the cell. Okay. So nothing inside over here, only the folding and the formation of class 2 MHC molecule is happening, that also within the endoplasmic reticulum. Now understand one thing, there is this invariant chain which plays a very important role here. As you can see, it is blocking the binding site of your class 2 MHC molecule. Why? Because this class 2 MHC is also present within your this area, right? Where your peptides are entering. Now, these peptides are not supposed to bind to your class 2, only to class 1. So to prevent that, to ensure that no peptides binds to your class 2 MSC, you have this invariant chain, and in this format only it is sent out. Okay, so MSC complex is routed through Golgi to endocytic pathway compartments. Okay, again it enters Golgi, then it gets into the endocytic. Oh, okay, never mind. It gets into this endocytic pathway where this invariant chain is degraded, leaving the clip fragmented. Okay, now you just have this small clip here. Now what happens? Your endogenous antigen is taken up, okay, through the endocytic pathway, and these vesicles they fuse. When they fuse, then these exogenous antigen, which by this time it has been degraded, okay, this gets loaded on the MSC class 2 molecule, and then it is subjected outside. Fine. So now think of the question. The question says that it is a virus who has sent its the proteins and the RNAs inside the cell, and now the viral peptide will be degraded, okay, and it will be presented. So it should follow which pathway? Clear about the question? So look at the question now. Now give me the answer. So Saritha and Rinku has already given me the answer. It is time for the others to give me the answer. I'm giving you 2 minutes time. Now it should be easy for everyone to give the answer once you have gone through the explanations, right? And I hope you have understood the explanation. I'm expecting some more answers. Navya is saying 3. Okay. So the correct answer is option 3. Very, very good. Now understand this. Here, even though they have said, see, they have made you like many people will get confused. They are saying presenting extracellular pathogen to cytotoxic T cell. So immediately you will think that, oh, they are talking about the exogenous antigen, right? But here this is a virus. Virus itself doesn't get inside the cell, it sends the protein inside the cell. So the protein, the viral protein, gets internalized and behaves as endogenous uh, antigen. Okay, so obviously it will be degraded and assembled by the class 1 MHC molecule. Okay, so this is a very simple question, but they have given a point where people can get confused. All right, otherwise it's a simple question. Okay, now moving over to question number 2. So apart from the interaction of antigenic peptide with TCR-CD3 complex signal 1, T cell activation requires another signal termed as co-stimulatory signal, signal 2. Given below are few statements regarding the co-stimulatory signals. Okay, so all three professional antigen presenting cells, dendritic cells, macrophages, and B cells possess the same capacity in respect to delivering the co-stimulatory signal. Co-stimulatory signals are not antigen specific. The principal co-stimulatory molecule expressed on antigen presenting cells are B7-1 and B7-2. Unlike B7, CD28, the ligand for B7, which is expressed on T cells, is not a member of immunoglobulin superfamily. Which of the above statements is or are not true? So this is a question which comes from the chapter of T cell activation. Okay, T cell formation uh, and activation. There you will get to see this particular picture. All right, where you can see the different fate of this APC and how it gets activated. So basically, this is telling you the interaction with your antigen presenting cell and the T cell. Okay, this is the antigen presenting cell and this is the T cell via your MHC complex, okay, major histocompatibility complex, and the T cell receptor. This is the T cell receptor. This is the MHC. You can see it, by, you can understand it by the structure, and this is a major MHC molecule. Okay, so your sorry, this is a T cell receptor. Okay, so MHC molecule has this antigen and it is presenting it. So this is the first signal that acts. Okay, it gives out the first signal. Okay, so this results in the energy of the T cell. Now what happens is this when the first signal is out, it will give out these fab anti 28 molecules. Okay, now initially they are like they will give out this molecule here, all right, which is going to be bound by your so this is your CD28 actually. This is the CD28, all right, which is initially blocked by this fab anti CD28, all right. So what is happening here, there is no connection is being is being formed. Okay, since it is blocked by fab anti CD28, so B7 is not being able to bind here. All right, so again you're going to get this anergic genes. Okay, that is the non-responsive genes. But when all the bindings happens, when the first signal is out, all right, the first signal is out, which gives out the second signal here. B7 binds to your this CD28 and this. So this is double signaling happening. Okay, so that results in the activation of your T cell. All right. So now you read the question and see what should be the answer. Okay, again, Sarita is saying A and D. Others, what do you say? Did you understand or you want me to explain once again? Have you all understood? If you have understood, then try to give me the answer. Okay, Rinku is also saying A and D. Others? So the statement they are saying number one is all three professional antigen presenting cells possess the same capacity in respect to delivering the co-stimulatory signal. Co-stimulatory signals are not antigen specific. The principal co-stimulatory molecules expressed on antigen presenting cells are B7-1 and B7-2. Unlike B7-12, CD28, a ligand for B7, which is expressed on T cell, is not a member of immunoglobulin superfamily. Which of the following are not true? Okay. Yugender is saying 1, Navya is saying B. Alright, the correct answer here is 3. Okay. Not true. All three professional antigen presenting cells possess the same capacity in respect to delivering the co-stimulatory signal. This is not true. Okay, it is different. They have different ways of presenting. Next, co-stimulatory signals are not antigen specific. This is true. Okay, the principal co-stimulatory molecules B7-1 and B7-2, this is also true. Unlike B7-CD28, the ligand for B7 which is expressed is not a member of immunoglobulin superfamily. That is wrong. The picture itself makes it very clear that it is a part of the immunoglobulin superfamily. Okay, so that is why A and D is not true. B and C is true. Okay, this is the structure itself gives you a very clear idea that it is your Ig superfamily protein. Okay, coming to question number 3. So in an experiment using nude mice, the population is divided into two groups, A and B. Group 1 mice are injected with T cells from normal mice, and group B mice are left untreated. Both the groups are then immunized with LPS. Which one of the following statements regarding antibody production in group A and group B is most likely to be true? So you can understand that there is two experiment given here. Okay. Now I don't have an explanation. I don't have any picture regarding this because this is an experiment. So you have to understand this. LPS lipopolysaccharide. Okay. So you have population is divided into two groups A and B. A is injected with T cell from normal mice. Okay. So this A group of mice, they're injected with T cells from normal mice. Okay. So I'm writing as normal. And group B mice, they are left untreated. Okay, that means no injection of your T cell has been given. Now what happens, both the groups are then immunized with LPS. So this group is also given LPS. Okay, and this group is also given LPS, right? Now they are saying which of the following statement regarding antibody production in group A and B is most likely to be true. So the first statement, they are saying that both groups of mice will have very similar levels of antibody against LPS, as it is a thymus independent antigen. No immune response will be generated in both groups of mice as they lack thymus. Only group A mice will have antibodies as T independent antigens are dependent on T cell activation. Only group B mice will have antibodies because in group A mice, the presence of T cell will interfere with the production of antibodies against T independent antigen. Okay, Sarita is saying polysaccharide, so independent. So Rinku is saying option 1. Yugender is saying option 3. Any other answers? Any other answers? Okay, Anil is also saying option 3. Okay, see, basically your thymus independent antigen is one immunogen which can stimulate your B cell to synthesize the antibodies. And for this case, you don't need the participation of T cells. Okay, so these antigens, they are very less complex than the thymus dependent antigens. So mostly these polysaccharides, they contain repeating epitopes, or you can say the lipopolysaccharides, they are derived from gram negative microorganisms, right? So this thymus independent antigens, they induce IgM mostly. IgMs which are synthesized by your B cells. And for that, obviously, you don't need any cooperation by your T cells. So these LPS, which activates the murine B cells without the participation by your T or the other cells, is typically your TL1 antigen. So even low concentration of your LPS can stimulate the synthesis of specific antibodies. All right. Whereas high concentration activate all the B cells to grow and differentiate. So therefore, the correct answer for this is option 1. All right. The correct answer for this is option 1. Both groups of mice will have very similar levels of antibody against LPS as it is thymus independent antigen. I hope you have understood, everyone. Okay, so you will get this topic under your antigen, the types of antigen, and B cell as well. Exactly, Sarita. Polysaccharide cross reactivity, high molecular weight cause B cell act without need of T cell. Right. Okay, now coming to your question number 4. Okay. So here they are saying that there are three substances A, B, C. Given below are the patterns of immunological responses in rabbits when number one A is administered along with C, B is administered along with C, and A is conjugated with B and administered along with C. Okay, so the first case A is administered along with C is this one. In the second case, you get to see that B is administered along with C is this one, and A is conjugated with B and administered along with C. So in your options, they have said which is what, like which is a protein, which is a hapten, which is adjuvant. So for this, you have to understand the definition of your hapten, adjuvant, immunogen. These definitions you need to know. Okay. So, what is your definition? So, if you see what is your definition, So if I first, there are a few things which you need to know. Okay, in your antigen presentation and this, what is an immunogen, what is antigen, what is a hapten, what is epitope, and what is adjuvant. Okay, so first understand that immunogen, okay, this is a stimulus which produces a humoral or cell mediated immune response. An antigen is that substance which binds to the antibody or a T cell receptor. Okay, so immunogens are substances which binds and it also initiate the immunological response. Antigen is a substance which binds. Okay, so I can say all immunogens are antigens, but all antigens may not be immunogens. Okay, now here comes the option. Some very small molecules can work as your haptens. Okay, that can bind to your antibodies or the B cell receptor, but they cannot initiate the immune response. Understood? So all antigens may not be immunogens. Why? Because some of them can become your hapten. So how will I define a hapten? So hapten is a molecule that reacts with specific antibody but is not immunogenic by itself. It can be made immunogenic by conjugation to a suitable carrier. Getting the point? Okay. So hapten. So you have antigen which binds to your antibody. Okay? Sorry, antibody B. If this elicitate immunological response, then you can call it as an immunogen. Okay, you can call it as immunogen. If it simply binds, okay, but does not initiate any immunological response, then it can be a hapten. Okay, so haptens are what? They are small molecules which can bind to your antibody, but alone they cannot initiate immunological response. How they can? If you add a carrier protein with them, then this conjugate can initiate the immunological response. Otherwise, not. Okay, so that is why, like, many drugs are hapten, like penicillin. It is a hapten. Okay, so it is an, you can also call it as incomplete antigen. All right. Now coming to your epitope. What is an epitope? So this is actually the antigenic determinant. Okay, so if you have this as the antigens, suppose this is your antigen. Okay, this entire antigen will not be able to initiate your immunological response. Only every antigen has an epitope. Okay, only this epitope is sufficient to initiate the immunological response. Clear? Now coming to your adjuvant. So basically, many a times what happens, if I want that drug to stay within the body for a very long time, okay, so that it can initiate more and more immunogenic response, then I use an inert chemical, okay, which will bind to the drug and it will keep it in the body for a longer period of time. Okay, so I can say that it is a pharmacological or immunological agent which modifies the effect of another agent. Okay, so maybe the antigen if I just give the antigen, it will only give me a very poor immune response. Okay, and it will be removed from very rapidly from the body. In that case, there is no point of giving that antigen, right? It won't be able to give me the what I desire, the amount of antibodies. So what I do, I add an adjuvant to it. Now you tell me which graph corresponds to which one. Think and tell me. Okay, Yogender is saying 2, Sarita is saying 2. Others, would you try? I hope everyone has understood the definitions of these. If you have any doubt, you can ask me. This is a live session, right? So you can clear your doubt. Why am I giving you this explanations and the definitions? Because many of you you skip this immunological part, this immunology section. But what I always suggest, like, don't skip it. It's not difficult. It's easy, provided you take it in a stepwise manner. You study it in a very stepwise manner. Don't jump from here to there. Then you will get confused, thoroughly confused. You have to first understand the definitions, and then you have to understand the mechanism. If that way you study, you will be able to understand immunology and answer the questions as well. Okay, it seems that no one else would like to try. So the correct answer is option 2. Okay, so as you can see here, they are telling you that A is administered along with C. Okay, and what did you get? So y-axis is telling you the antibody response, that is immunogenic response. You didn't get any response, right? So likely A is a hapten. Okay. Next, what you see, A is administered along with C. And when A is administered along with C, you get a response here. So that means what? This C must be some protein, some carrier proteins. That is why you got a response, because hapten, when it is conjugated with carrier protein, it gives you response, right? So B is a protein, okay, or a carrier. Next, they are telling you that A is conjugated with B and administered along with C. And now what you see, you get to see a response for A, and you also get to see a response for B. Okay. So A is conjugated with B and administered along with C. So A and C obviously is going to give you this, right? But when you are also having this B and C, no? Yeah. So then C becomes the adjuvant. All right. So answer 2. So this is the thing. You have the carrier protein, hapten. This will give you the result. Now, if you immunize, you will get to see antibodies which are formed specific to the hapten. You will get antibodies specific to the carrier proteins. You will also get to see antibodies to get in conjugate with like both of it. Okay, hapten and carrier. An adjuvant for the prolonged persistence, enhance co-stimulatory signals, increase local inflammation, and stimulate non-specific proliferation. Fine. Now, coming to the last question. So, it is saying that the immune system of thymectomized and lethally irradiated A into B F1 mice H2AB were reconstituted by grafting thymus from strain B mice H2B and with A into X F1 bone marrow cells. These mice were then infected with lymphocytic choriomeningitis virus. The CD8 cytotoxic T lymphocyte from the reconstructed mice were then co-cultured with the following cells. Which of the proteins below indicate the cells that will be lysed by CD8 cytotoxic T lymphocytes from the reconstituted mice? Read this very carefully, step by step. If you have to, then do some scribble work. So, what it should be? Okay, take your time. So, so this is talking about the CD8 T cell target recognition, right? So your CD8 T cells, they recognize and kill the virus infected cells, which are presented by your viral peptides on MHC class 1 molecule. So what they have given you something regarding your strain B H2B. Okay, so you want me to give the answer? So see, here since the thymus is from your strain B, okay, strain B mice H2B, and the CD8 T cells, they will be restricted to your H2B molecules. right? So the reconstituted immune system, it actually has originated from your A into B F1 bone marrow cells. Just a second. Okay. So now that means it can generate the antigen presenting cells with both your H2A and H2B. Okay. Now, this T cell selection, it only occurs on your H2B MHC. That means the cytotoxic T cells will primarily recognize H2B restricted targets. Okay. At the same time, you will see that the CD8 T cells, they will not attack the uninfected cells regardless of your MHC type. All right. So therefore, in this case, the correct answer is option 3. Okay. Because now if you see the infection, okay, so this LCMV, this was infected fibroblast. All right. So in the first case, you will see this AB F1 infected fibroblast. Let me write it down. This one. Okay. AB F1 infected fibroblast. Here you will see the H2A, H2B present. Okay. So that contains H2B. So therefore, what will happen? It will be lysed. Okay. Now next you have strain A infected fibroblast. All right. Strain A infected fibroblast. It has your H2A only, so no H2B, so it will not be lysed. All right. In the next case, you see strain B infected fibroblast. This one. In this case, you have H2B only. So it only contains H2B, so it will be lysed. All right. So in this case, D and F will be lysed. Understood? Understood, everyone? I'm not getting any responses. Am I audible? All right. So these were the 5 questions that I had for today's discussion. I hope you have got an idea as to what kind of questions comes and how you should prepare. Okay. So don't just study the theory, but also study the applied whenever you're thinking of like this is happening, this reacting, this is happening. Think of it in the other way. What if it doesn't happen? Okay. Only then you will be able to answer these kinds of questions. So once you finish revising, you can go through this lecture once again, try the question yourself once again, and also you can take up or practice more such questions in our T3 test series. Okay, so you have to download the Biotecnika app over there. This series is given free. Okay, so you can go through this. And for those who want a detailed explanation from us, you can definitely reach us through the toll free number and get enrolled in the Lakshya batch. So that's all for from my end today. Thank you everyone and wish you all the best. Thank you.